So our sarcomere has gotten shorter. All right. Now let's go ahead and look at this I band. All right. We only were looking at half of it, but here we see that it covered the distance from here to here. Let me go ahead and get a different color marker here. Here we see that that I band covers from here over to here. Now if we draw this straight down, we draw there, we see that the I band also gets shorter, right? What about the A band, that dark band? Okay, well, I did my best to try to stay as consistent as possible here, but you'll notice that the A band, well, more or less stays the same, all right? The A band more or less stays the same. And then we notice that the H zone, which is this zone right here, obviously gets shorter, right? So the sarcomere gets shorter, the I bands get shorter, the A band stays the same, and the H zone gets smaller as well. So what this told early anatomists is that somehow there's some mechanism that when a muscle shortens, those thin filaments are sliding past those thick filaments. All right, so that's what we want to look at next, is we want to look at the mechanism of how muscles shorten. So let's go ahead and do that now. Let's go ahead and look at how muscles shorten. Right. How does a muscle shorten? There are going to be three basic things that happen, three different phases that we're going to be talking about. Right? The first thing that we're going to be talking about are the events that take place at this anatomical structure called the neuromuscular junction. So the events at the neuromuscular junction. All right. The second thing that we want to talk about is we want to talk about what we call excitation-contraction coupling. All this is really saying is, how are we coupling the excitation of the muscle with the actual contraction? So what's happening in the muscle to essentially propagate the signal that we're starting? And then the third thing that we have to deal with is we have to deal with the actual contraction itself. Okay, and this is called the sliding filament theory of muscle contraction. All right, so we have these three kind of separate but continuous events that are happening in the muscle that allow it to shorten. So let's go ahead and start with number one, and let's go ahead and take a look at how a muscle itself looks. So we're going to have to back out, right? We started with a muscle cell, we looked at myofibrils. Now I want to back out and I want to look at a muscle itself. All right. So skeletal muscle, all right, so skeletal muscle itself is so named because it's attached to our bone. So here's our bone right here. And we have a muscle attached to it via a tendon, right? So we have this muscle that's been attached. Okay, we're kind of cutting off the other end of it here. So we have this sort of tendon, right? And then we have the muscle itself. So it kind of looks like that. We're going to cut that muscle like this, okay? So the muscle itself is, has these fibers associated with it, and there you have it, my poor artwork, all right? And then the muscle is made up of multiple muscle fibers, okay? tendon right here. We have the muscle. And we have hundreds of muscle cells that make it up. Right? Now, when we talk about contraction, and we're talking about these series of events, we are going to be looking at a single muscle cell. All right? So we're going to go through that process. We're going to talk about how one of these single muscle cells contracts. And then we're going to talk about how an entire muscle functions, because there are some differences and some nuances when we talk about that. All right? Each one of these muscle cells does have the ability to contract when it is stimulated. Okay. Now, you guys have probably been to the chiropractor or the doctor or the physical therapist or something like that, where they put a muscle stimulator on you. And what those do is they apply direct electrical current to your muscle cells, and it causes them to contract, right? You can know what that feels like. But in your body, you don't have a muscle stimulator that allows you to do that. What you have is you have neurons. All right, so we want to go ahead and talk about these neurons that are part of our nervous system and talk about how they work to control these guys. All right, so what I want to do is I want to go back and I want to look at a couple of muscle cells. So we're going to blow up some muscle cells here, and we're going to take a look at how neurons work to stimulate them. So here's my muscle fiber. Muscle cell, right? Okay, so this is my muscle cell. And there are neurons coming from our central nervous system that are going to control or stimulate or innervate our muscle cells. These cells are called motor neurons. Right, so here's a cell body of one of these neurons that we have right here. And then the axon of this guy is going to run all the way down. It's eventually going to encounter our muscle cell right here. We're going to talk about neurons in a lot more detail, right? But what we want to do is we want to go ahead and go over some basic things, right? This is called the cell body, or soma. Of the neuron. Okay, there's the nucleus that we have right there. All of these little projections that I've drawn all over the place here, these guys are called dendrites. And then most neurons have a single long projection called an axon that is going to travel away from it and go to some other organ. In the case of these motor neurons, that axon is going towards this muscle cell. All right. Now, this axon can branch. All right. This axon can have branches that kind of come off of it, and those branches might go to other cells in the area. All right. So we might have another muscle cell kind of tucked up right here next to this guy right here, and this other axon could potentially be going to it, right? So we might have multiple muscle fibers here sort of in line with each other, and this axon could go to multiple muscle cells. All right? When we have one muscle cell plus all of, I screwed up. I'm really sorry about that. We have one motor neuron plus all of the muscle fibers that it innervates. We call that a motor unit. Right? We call that a motor unit. So one motor neuron plus all of the muscle fibers it innervates is called one motor unit. So what we've drawn in here is essentially a motor unit. We've drawn a single motor neuron, and we've drawn two muscle cells that it's going towards, right? This would obviously be a very small motor unit, okay, but we do have small motor units in our body. There are some muscles in our body where we have really, really fine motor control, where the motor units are quite small, potentially like this one. But then we have other muscles in our body where the motor units might be a lot bigger, right? Like think about the big, powerful muscles in your thighs and your arms and stuff like that. You know, those motor neurons could be going to hundreds of muscle fibers where you have a lot of strength, but you don't have as much motor control. But we'll elaborate on that in just a little bit, right? But it's important to remember that the axon can branch, and that axon can go to multiple muscle fibers. Now, when that axon stimulates, or when that neuron stimulates these muscle fibers to contract, they're all going to react the same way. So this one right here would react the same as this one that we see. All right? The end of the axon is called the axon terminal. Okay, the end of the axon is called the axon terminal. So this is what we have. The cell body is going to be found in our central nervous system, and these axons are packaged as part of the nerves that run through our body that go towards our muscle cells. So an individual muscle would have multiple axons running towards it and bundled into these nerves. All right? What we're interested in right now is we're interested in what's happening at this place right here, where we have the axon meeting up with the muscle cell. This particular area right here is what we call the neuromuscular junction. All right, the neuromuscular junction. It's simply called that because it's where we have the nervous system and the muscular system coming together, right? So we want to look first at what's happening in terms of muscle contraction at the neuromuscular junction. Let's go ahead and do that. We're going to blow up that neuromuscular junction and kind of see what happens next. So I'm going to go ahead and draw my muscle cell again right here. And I'm going to draw my axon coming in here kind of like this. So there's my axon. And there's my axon terminal, right? So this is my sarcolemma right here. This is my axon. And this is my axon terminal. So we have this. All right, so we've just simply blown up what we had in the last slide. Okay. Now the message that is going to eventually tell this muscle cell to contract, this message that originates in the neuron, is called an action potential. All right, it's called an action potential. An action potential is an electrical signal. We're going to go into action potentials in a lot of detail in the next unit of study, but suffice it to say right now, it's an electrical signal. There are ions, sodium and potassium ions, flowing across the membrane of the axon, and what you have when you have ion flow is you have something called an electrical current. This electrical current that we find in axons as an action potential is being propagated is in a lot of ways no different than the electrical current that you find in your house, right? And you can measure that current in volts, all right? So we're going to be measuring action potentials and action potential propagation in millivolts this semester. You're going to be doing that in lab. In your house, of course, there's a lot more ions flowing than there are in your body, so we measure that in volts, not millivolts. But at any rate, it's the same basic idea. So an action potential is very much an electrical signal. And that action potential is traveling down the axon at a very high rate of speed. All right? So that electrical signal is getting propagated down the length of the axon, and eventually it's going to reach the axon terminal. Okay? When that action potential reaches the axon terminal, what it does is it triggers calcium channels to open on the surface of the axon terminal. So the membrane here is going to have all these calcium channels. And they're normally closed, but when the action potential reaches the axon terminal, those calcium channels open. Okay? So we're going from a closed state to an open state. All right, let's go ahead and write these things down. So we're going to say here number 1, and then we're going to say number 2. Right? So number 1 is that an action potential travels down the axon and reaches the axon terminal. Right? Number 2, right, that we have right here is voltage gated calcium channels open. Right? Voltage gated calcium channels open. They're called voltage gated channels because they're opening in response to this change in voltage, this electrical signal that we have here. All right? They're calcium channels, and calcium is found in high amounts outside of the cell. So when these channels open, okay, and there's all of a sudden a passageway for the calcium to move, it's going to diffuse into the axon terminal. Okay? So calcium diffuses into the axon terminal. All right, and that's what we have happening there. Okay. The neuron has also been making a substance called acetylcholine. Okay, these neurons are constantly making this substance called acetylcholine, and they're packaging this substance into vesicles that we find in the axon terminals. So we have a number of these vesicles here that are containing this substance, acetylcholine. Acetylcholine is a neurotransmitter. It's going to send a message to the next cell in line. And in this instance, that next cell in line happens to be the muscle cell. So these are vesicles with acetylcholine. I'm going to abbreviate that as ACH, right? So these are vesicles with acetylcholine. And those are found here in the axon terminal. What's going to happen is that when calcium rushes into the cell, it triggers the exocytosis of these vesicles. So what's happening here is that we have exocytosis occurring. We have these vesicles fusing with the membrane of the neuron, and the contents of those vesicles are going to be dumped into this space. Okay? Anytime we have a junction between two cells, that's called a synapse. And this particular space right here is called the synaptic cleft. So what we're doing here in this next step, we're going to label that number 3, is we are emptying acetylcholine into the synaptic cleft. So number 3, acetylcholine enters cleft, okay, via exocytosis, okay, and well, what is the acetylcholine going to do? It's going to start to diffuse into this space, right? It's going to diffuse into this space. So we have acetylcholine diffusing throughout this space, right? We dump it in there and it starts to diffuse. And what it's going to do is it's going to start to bind to receptors that we see on the sarcolemma, on the muscle cell membrane. On the muscle cell membrane, there are acetylcholine receptors, right? And we see them present mostly in the synaptic cleft. So when it diffuses into this really small space, the acetylcholine is able to bind to receptors, okay? So we're going to say that acetylcholine enters the synaptic cleft via exocytosis and binds receptors on the sarcolemma. Okay, it binds receptors on the sarcolemma. Okay, so what happens to those receptors? What's going to be occurring when that acetylcholine binds to those receptors? Well, when acetylcholine binds to these particular receptors, what it does is it opens those receptors up. They're big channels, very much like these calcium channels that we saw on the neuronal membrane. All right? However, instead of them being permeable to calcium, these receptors are going to be permeable to sodium and potassium. All right? So when the acetylcholine binds to these receptors, what happens is sodium and potassium start to flow across the membrane of the muscle cell. Now, we find the majority of our sodium, outside of the muscle cell. So sodium is usually floating around out here in the extracellular space. And we find the majority of our potassium inside the muscle cell. So potassium is floating around here in the intracellular space. So when acetylcholine binds to these receptors and these receptors open, what's going to happen is that sodium is going to diffuse into the cell and potassium is going to diffuse out of the cell. All right, so we're going to see a movement of potassium out of the cell, and we're going to see a movement of sodium into the cell. So we're going to see something that sort of looks like this, a movement of these. Now, sodium and potassium are electrically charged. They happen to have the same charge, but they are electrically charged. They're ions. And when that happens, it changes the electrical balance of the muscle cell. And it starts an electrical signal called an action potential in the muscle cell. All right? So what we've done here is we had an electrical signal, the action potential, that traveled down the motor neuron, the axon of the motor neuron, until it reached the axon terminal. We converted We converted the electrical signal into a chemical here at the synaptic cleft, right? This chemical signal was an acetylcholine, and that acetylcholine was able to start an electrical signal here at the sarcolemma. So we've gone from an electrical signal to a chemical signal back to an electrical signal in the muscle cell. All right? These channels open because of the binding of acetylcholine. We call them chemically gated channels because a chemical bound to them. But that did start an action potential in the muscle cell membrane. All right? So this series of events at the neuromuscular junction is what's going to trigger this muscle cell to contract. The action potential travels down the motor neuron, opening these voltage-gated calcium channels. The entry of calcium into the cell is going to trigger the exocytosis of these vesicles, containing this neurotransmitter acetylcholine. Acetylcholine fills the synaptic cleft, and it binds to the acetylcholine receptors on the sarcolemma, and sodium and potassium then can flow across the muscle cell membrane, starting an action potential in the muscle cell. All right. So what we want to do next is we want to look at this so-called excitation-contraction coupling and see how that action potential is propagated in the muscle cell and what effect it has on the muscle cell. All right.